Okay, folks, so the hard part of chapter 25 is over. That was the 25-2, the control mechanisms of all those metabolic pathways. So now we're finished with carbohydrates and some of the central metabolic pathways. And now we start with the lipid metabolism. I don't believe this picture's in your book. It's only here to show you one thing. Like carbohydrate metabolism, where I showed you glucagon and epinephrine, hormones control lipid metabolism as well. So what you see here is a hormone, the hormone, the ligand. Remember, a generic term for something that binds a receptor is ligand. The hormone's the ligand. You see the ligand or the ligand binding the receptor, turning on adenylate cyclase and making the second messenger cyclic AMP. You're probably really very used to that by now. Just like the control mechanisms in carbohydrate metabolism, where phosphorylation was a trigger, sometimes triggered it on and sometimes triggered it off, but phosphorylation was a trigger, it's the same thing. We activate a protein kinase and we trigger the phosphorylation of an enzyme. In this case, lipase. Lipase is inactive when it's dephosphorylated and it's active when it's phosphorylated. So now lipase is phosphorylated, and uh, lipase breaks down triglycerides, also known as triacylglycerols, into glycerol plus three fatty acids. One, two, three. Because here's the three fatty acids that were here. One, two, three. So now I have a glycerol and three fatty acids. Now what happens is the glycerol is run through glycolysis. It enters glycolysis. And the fatty acids go through another metabolic pathway called beta oxidation. So that's hormone control of lipolysis. This is called, this is called lipolysis or lipolysis, breakdown of fats. Here's your triglyceride right here, your triacylglycerol or triglyceride. This enters the cell and is broken down into glycerol plus three fatty acids. There's actually three of these because there's three of these here. And these fatty acids enter the mitochondria. Now, there's a transport mechanism involving carnitine to get these fatty acids into the mitochondria. It's a pretty cool transport mechanism. We'll look at it briefly. As soon as the fatty acids are in the mitochondria, they go through this thing called beta oxidation. That's this word right here. And why it's called beta oxidation, and look at this purple box right here, is because you're breaking two carbons off at a time. So it's a two-carbon oxidation pathway, so we call it beta oxidation. And those two carbons are attached to CoA, and they make up what we call acetyl-CoA. That probably sounds familiar to you because pyruvate was converted to acetyl-CoA in the carbohydrate metabolism. Well, fatty acids are beta oxidized into acetyl-CoAs. And the acetyl-CoA just enters the Krebs cycle just like it did before, the citric acid cycle. So that's fat metabolism. Now the thing here is, depending on how long these fatty acid chains are, this one's an 18 carbon fatty acid chain. So if you take two carbons away at a time, you get 9 acetyl-CoAs. You'd get 9 of these. But that's for one fatty acid. There's 3 of them. And if all 3 of them are 18 carbon fatty acids, and you get 9 acetyl-CoAs per, well, that's talking about 9 times 3 equals 27. That's 27 acetyl-CoAs. Now think about that. A glucose molecule gives you 2 acetyl-CoAs. A glucose molecule is broken down into 2 pyruvates in glycolysis. Each pyruvate is converted to acetyl-CoA. So a glucose molecule gives you 2 acetyl-CoAs. One triglyceride gives you 27. So you can see that one triglyceride holds much more energy than one glucose molecule. All right, so that's beta oxidation. This is showing you that when I do make a bunch of acetyl-CoAs, well, please don't memorize this metabolic pathway here. It came from a biochemistry textbook. Here's acetyl-CoA. That's what this is. It's acetyl-CoA. When I do a lot of fat metabolism, I make a ton of these acetyl-CoAs, just lots of them. because you can see that I made 27 from one triglyceride breakdown. So you make a lot of these. Well, when I have a lot of these start building up and I'm overwhelming the citric acid cycle, so I can't funnel them through the citric acid cycle fast enough, they start to be converted to other things. Here are the enzymes, and please don't memorize the enzymes by any means. But when they're converted to other things, what I get is the three major ketone products: acetoacetate, acetone, and beta hydroxybutyrate. I would like you to know these three major ketone products because these three ketones lead to ketoacidosis. Because acetoacetic acid, beta hydroxybutyric acid. So I get, and then acetone is doesn't deprotonate like these other two, but these are three ketones, three major ketone bodies, and they lead to a thing called ketoacidosis. So I would like you to memorize these three ketones: acetoacetate, acetone, and beta hydroxybutyrate. All right. And the three ketones will give us ketoacidosis. So the question is, when do I have a predominance of lipid breakdown? And the answer is when you're not using glucose. Your cells have to burn something. So if they're not being allowed to burn glucose, they're going to burn the fats, and you'll get ketone bodies. So then the question is, well, why is it I can't use glucose, assuming I'm eating plenty of it? And the answer would be when you're diabetic, when you don't have the insulin to tell the cells to absorb glucose. So one reason for ketoacidosis is diabetes. This is the transport of the fatty acids across the mitochondrial membranes. First of all, this one is a simplified. Let me do this one first. It's simplified. This one is not as accurate as the previous one, but it's the general concept is the same, just so you know. All right. So first of all, what I have to do is I have the fatty acid CoA. The fatty acid is attached to a coenzyme A, and it's called an acyl-CoA. Well, what happens is a carnitine molecule replaces the CoA, and I get an acyl-carnitine. This is transported into the mitochondria. All right. Now, I have one transport molecule that gets it into the intermembrane space. This is the intermembrane space right here. And then I have another transport molecule that gets it into the matrix right here. So the acyl-carnitine gets into the matrix here. Well, this picture is not exactly true, but the gist of it is, after the acyl-carnitine is in the matrix, I reverse the original reaction. A coenzyme A replaces the carnitine, and I get my acyl-CoA back that can do beta oxidation. And then the carnitine can go back out and be used again. So the carnitine goes back out. All right. And actually, that shows you that right here. Here's where it shows you the carnitine going back out. So it sounds a little foolish to replace the coenzyme A with carnitine just to replace the carnitine with coenzyme A later. But it's necessary to get the fatty acid into the mitochondria. So that's what's going on here. And I actually have two different carnitine palmitoyl transferase enzymes that do this. I have the carnitine palmitoyl transferase enzyme that attaches the carnitine to the fatty acid to get acyl-carnitine. And then I have the carnitine palmitoyl transferase that replaces the carnitine with coenzyme A. And you can see they're red and they're blue. All right, now this picture, it's a decent picture. The concept is correct. One of the details is not. This is more accurate. Here I have my fatty acid. By the way, the R group means it's a fatty acid, but it could be an 18-carbon fatty acid. It could be a 16-carbon fatty acid. It could be a 14-carbon fatty acid. So they just put the R group there saying, hey, we don't know how long the fatty acid is, but it's a fatty acid. The fatty acid is attached to a coenzyme A. Here's my coenzyme A. and it's called acyl-CoA. Acyl-CoA enters the intermembrane space, and the carnitine palmitoyl transferase enzyme replaces the CoA with the carnitine, and I get acylcarnitine. Now, the acylcarnitine enters the matrix, and I get another carnitine palmitoyl transferase enzyme that replaces the carnitine with CoA, and I get my acyl-CoA again. So when I started out with acyl-CoA here, I end up with acyl-CoA here. But I had to go through this acylcarnitine intermediate just for transport purposes, and that's it. So these carnitine palmitoyl transferases or carnitine acyl transferases, they're very important in transferring the fatty acid into the matrix of the mitochondria. And then, of course, the acyl-CoA just goes through beta oxidation. And if it's 18 carbon fatty acid, you get 9 acetyl-CoAs. If it's a 16 carbon fatty acid, you get 8 acetyl-CoAs, etc., etc. So there you go. That's the carnitine transport mechanism. You might have seen carnitine in your vitamin section of the store. Now you know what carnitine does. All right, these next couple slides are not in your text, and they're not for you to memorize. They're just to explain where we get the nomenclature. So first of all, lipids are hydrophobic or lipophilic, however you want to say it. So lipids are hydrophobic or lipophilic. And that means they don't float around the bloodstream very well because the plasma is 90% water and lipids are hydrophobic. So they're carried by proteins, and these are called lipoproteins, but there's different lipoproteins. We have the lowest density lipoprotein called chylomicrons. Density a lot less than water, less than 1. Water's 1. Water is 1 gram per milliliter. That's water. And by the way, the other way of writing that is 1 gram per milliliter to the negative 1. That's just the same way of writing gram per milliliter, just so you know, and that's how it's written here. It's just don't worry about that. It's like writing gram over milliliter. So chylomicrons has a density less than water. The very low density lipoproteins has a density around water. Sometimes a little less, sometimes a little more, but right around water. You can see that when it's a little bit more, it's very little bit more, because water is exactly 1. Then you have intermediate density lipoproteins. Then you have low density lipoproteins, and this is what's called the bad cholesterol. Cholesterol is cholesterol. There really is no such thing as bad and good cholesterol, but we'll talk about why we call LDL the bad cholesterol and why we call HDL the good cholesterol. And again, remember, cholesterol is just cholesterol, but we'll talk about this. All right, so those are the different lipoproteins from chylomicrons to HDL. Same thing, but showing you the constituents. And if you look at the key over here, the yellow is a triglyceride. That's gray, that's brown, that's purple. So yellow, gray, brown, purple. Brown is cholesterol. I'm not going to pay attention to phospholipids much at all. I mean, they're part of it, but I'm not going to pay much attention to it. I'm really going to pay attention to triglycerides and cholesterol and protein, by the way. Let me remind you about something. When you have a protein that hasn't bound its prosthetic group, it's called an apoprotein. And then when its prosthetic group is added, it's called a holoprotein. Okay, I'm just reminding you because I'll come back to that. Well, if I look at chylomicrons, it's 80 to 95% triglycerides. And you know, in your Italian dressing at home, you know that the fats float on top of the water. You know that. And you have to shake it up to get them to mix before you pour it on your salad. So these chylomicrons have a very, very low density because they're predominantly triglycerides. All right, and you can see that they don't have much protein in them. Well, the next lipoprotein is called very low density lipoprotein, VLDL, very low density lipoprotein. Less triglycerides, more cholesterol, and more protein. Oh, I'm sorry. When I said cholesterol, I pointed at that. Here's cholesterol. So I went from 2 to 7% cholesterol to 10 to 15% cholesterol. So I have more cholesterol. I have less triglycerides. I actually have more protein. But still a low density. A very low density. Let me be precise here. Notice that the VLDL is made in the liver. All right, the next one, low density lipoprotein. Far less triglyceride. Far more cholesterol. Matter of fact, the LDL is the one that has the most cholesterol. The most cholesterol. But that's not even what makes it bad cholesterol. I mean, we're going to call this bad cholesterol, but cholesterol is just cholesterol. That's not what makes it bad, having 45%. Something else makes it bad, and I'll talk about it in a second. And you can see that the LDL has more protein as well. All right, now HDL. This is the good cholesterol. Now, cholesterol is just cholesterol. There's nothing good or bad about it, but why do we call HDL good? Because of something I'm going to talk about shortly. Far more protein, so it has a higher density. This is the highest density lipoprotein. Far more protein here. All right, now let's talk about what makes these bad and good. LDL is made in the liver and sent out to the tissues. That means LDL is sending out into the bloodstream to go to different tissues. And it's the LDL that can cause atherosclerotic plaques because we keep adding LDL to the blood to go out to the tissues. I mean, your bloodstream is just a transport medium. So when the LDL is added to the blood to go to some tissue someplace, it can be embedded into my blood vessels and cause atherosclerotic plaques. So LDL is going out into the body to other tissues and being transported by the blood, so it could clog up your blood vessels. Whereas HDL is being returned to the liver. Now that still has to go through the bloodstream, but my liver is clearing it. My liver is gobbling it up and destroying it and adding the cholesterol to the bile. So my HDL is really clearing the cholesterol from my bloodstream. It's taking it out of the bloodstream and taking it to my liver. And in addition, that's why it's called good cholesterol. And in addition, HDL floats around with far less cholesterol than LDL. So it's the direction that the cholesterol is going, really, which makes it bad or good. Now, this is a picture from a biochemistry book. You do not have to memorize all of this, except for I would like you to memorize two things. These, well, actually, ApoB100 is the most important one. I'll only have you memorize one of them. There's two of them that are important here for what I want to talk about, but let's just memorize one of them. ApoB100 is the protein part of the lipoprotein that you find in LDL. In fact, ApoB100 protein binds the LDL receptors. Now that's pretty cool. All right. Now think about this. Because what happens is we internalize LDL into our cells that are going to use it by receptor-mediated endocytosis. And you probably remember that from Bio 203. Receptor-mediated endocytosis. But what would happen if I didn't have the receptor? Your LDL would not be cleared from the blood. It would stay in the blood. It would not go to its target tissue. Oh, that's not good. LDL is a ton of cholesterol, and that's staying in my bloodstream now. Yep. What would happen if I didn't have ApoB100? What would happen there? And the answer is, or if it was I had the ApoB100, but there was a mutation in the gene, therefore the protein is not functioning well. It's not recognizing the receptor. LDL would not be internalized, and it would stay in the blood. So you can see that if I have a problem with ApoB100, or I have a problem with my LDL receptor, I get hypercholesterolemia because my LDL stays in my blood. Cholesterolemia.
So these two proteins are important, and I can get familial hypercholesterolemia, meaning it's genetic if I have a problem with my ApoB100 protein or my LDL receptor. All right. This is showing you the LDL. How do I know it's LDL? Because it's ApoB100. And it's showing you that the ApoB100 carrying the cholesterol binds its receptor right here. It's internalized in this clathrin-coated pit. Here's it's in a coated vesicle. I recycle my receptors. Here's recycling my receptors back to the membrane. And I digest my LDL. I recycle my amino acids, but my cholesterol becomes part of bile. I put it in my bile. Well, that's only if it's the liver. If this is happening in the liver, I put it in my bile. But if this is happening in my muscle, it's used for something else, like maybe membrane maintenance or something like that. So the cholesterol is being used. It could be being used for steroid hormone synthesis. Cholesterol is actually the precursor of all steroid hormones. So whatever the cholesterol is being used for, it's being used here. Notice that I have two sources of cholesterol. Notice I have an endogenous source of cholesterol. That's cholesterol I synthesis of cholesterol. And notice I have an exogenous source of cholesterol. Cholesterol I ate in my diet. But this cholesterol pool right here comes from two sources, endogenous and exogenous. Well, what happens if I want to lower my cholesterol because it's too high, my blood cholesterol is too high? Well, I have to reduce both sources of cholesterol. I have to reduce my cholesterol synthesis, and I have to reduce my cholesterol in my diet. Well, reducing the cholesterol in my diet is up to me. It's a behavioral change. It's just me making decisions in the food I eat. But how do I tell my cells to make less cholesterol? Well, that's a little trickier. It's not like I can think my cells into making less cholesterol. But I do have drugs I can take, and the drugs we take inhibit this enzyme right here, hydroxymethylglutaryl-CoA reductase. I have drugs that knock out that enzyme. If I knock out that enzyme, I don't synthesize cholesterol endogenously. And all I have to do now is reduce my dietary intake of cholesterol, and I can have an overall lowering of my cholesterol. All right. So that's endogenous and exogenous cholesterol. Well, where did my exogenous cholesterol come from? It came from lipids I ate that bile emulsified into a mycel. I absorbed my mycel, and then I dumped those lipids into my lacteals of my lymph system. They're always carried by a chylomicron first. First, they're carried by a chylomicron. Now, what happens is when the chylomicrons, because the lymph fluid is ultimately dumped into your bloodstream in the right thoracic duct the right lymphatic duct in the left thoracic duct. Remember the left, it's actually just called the thoracic duct, but it's on the left. The left thoracic duct in the right lymphatic duct. They dump lymph into my subclavian vein. I don't know if you remember that from Bio 203. But ultimately, my lymph is dumped into my blood. And then it's flowing in my blood. Now, what happens is when it gets close to its target tissue, like muscle or adipocytes, or it could be the adrenal cortex, because the adrenal cortex makes a ton of steroid hormones, or it could be the ovaries, or it could be the testes. Whatever its target tissue is, when it gets close to there's a lipoprotein lipase in the endothelial layer of my blood vessels. And this breaks down the chylomicrons and releases the fatty acids and the glycerol to my tissues, my skeletal muscle, or my adipocytes, or my ovaries, or my testes, or whatever. All right, and then these tissues absorb those lipids, whether it be monoglycerides and fatty acids or cholesterol, and they utilize them. That's what they do. They utilize them. Now, adipocytes store them. Skeletal muscle will store fats a little bit, but it burns them as well. So you can see that going on here. I eat my lipid, I absorb my lipid, it's transported via chylomicrons to the target tissue, and then broken down by lipoprotein lipase, and my target tissue absorbs them. Okay. Meanwhile, all right, meanwhile, I have HDL picking up any extraneous lipids and taking it to the liver to be cleared. Meanwhile, if I'm running short on lipids, I have LDLs from my liver going out to all tissues delivering them. So you can see that I have this transport mechanism for lipids. And this is showing you if I need to send cholesterol to a tissue or triglycerides, I send it via this LDL. If I have excess cholesterol in my tissue, I grab it with HDL and I send it to my liver. Now, here's the question. What happens to the cholesterol in my liver when I have it, when it's absorbed there? I put it into my bile. I put the cholesterol into my bile. And that goes down into my intestine and helps me digest more fat. All right. Now, there's cholesterol lowering drugs out there that are bile sequestrants. Now, just remember, you just did this chapter a few chapters ago, one chapter ago. Sequestration. You know that the reason bile is made and released to the duodenum is because cholecystokinin told the liver to do that. It said make bile and release bile. And you know that cholecystokinin is secreted from the duodenum when you have lipids in your duodenum. Now think about this. What would stop that cycle of cholecystokinin saying to make and release bile? Reducing the lipids in your duodenum. Well, what reduces the lipids in your duodenum? Bile emulsification of the lipids in my duodenum and absorption of those lipids. But what if I take a drug that is a bile sequestrant? It sequesters or chelates bile and doesn't allow it to go to the, well, it allows it to go to the duodenum, but it sequesters it and doesn't allow it to do its job. Well, then the lipids stay in the duodenum longer, which leads to more CCK, which leads to more bile production. But that bile is not functional because I have a drug in my system that's a bile sequestrant. So in other words, these bile sequestrants cause me to make a bunch of bile. Well, what's put in bile? Cholesterol. So the more bile I make, the more cholesterol I put into it, the less cholesterol that's in my blood. So some of these drugs are bile sequestrants and they lower my blood cholesterol content. All right. This is the collage showing you absorption of the lipid from the intestine, its transport through the lymph system to the bloodstream, its delivery to the target tissue, and then these cycles here where LDL takes cholesterol and other lipids out to the cells of the body, and HDL clears cholesterol from the body and delivers it to the liver. So this is a cycle going on. Now, this is the endogenous production. This is the endogenous production of cholesterol. Endogenous cholesterol. This is not the stuff you ate. It's up to you and me to reduce the cholesterol we eat. This is the endogenous production. Please don't memorize the pathway. Basically what happens is you start with acetyl-CoAs and you make cholesterol. That's basically what happens. All right, here's cholesterol. Now, there's an enzyme right here. It's a critical step in this pathway. It's controlled by hormones. See? That enzyme can be inhibited by drugs. Well, first of all, you know negative feedback. If I have high mevalonate, that's going to negatively feedback and shut down the enzyme that made it. That's what it's going to do. There's negative feedback on this. All right? Feedback inhibition or negative feedback. That's how we control the production of endogenous cholesterol. Well, I also have drugs, like these drugs right here. These drugs right here have a mevalonate type moiety. See, it looks like that. It looks like mevalonate. So Lipitor, Mevacor, and all these statin drugs, they have a moiety that looks like mevalonate. And therefore, these drugs inhibit hydroxymethylglutaryl-CoA reductase, and you don't make endogenous cholesterol. And then all you have to do is lower your cholesterol in your diet, and you're good to go. Okay, folks, that's it for the lipid metabolism.